Welcome to this explainer. You know, the history of biology is, well, it's essentially humanity's epic attempt to clean up a wildly messy, ever-expanding room. I mean, for millennia, we've tried to shove nature into neat little metaphorical boxes. But as our microscopes got better and our tools evolved, we quickly realized this room was practically infinite. So today, we're going to explore the brilliant and, honestly, sometimes chaotic history of biological classification.
Okay, let's dive into this. It's the ultimate puzzle, right? Think about it. If you were handed the impossible task of sorting every single living thing on Earth, from a towering redwood tree down to the microscopic bacteria on your skin, where would you even begin? That is the foundational question that's literally driven scientific observation since the dawn of civilization. Now, our earliest attempts were totally instinctual. We just grouped things by how we use them, like food, shelter, clothing. But Aristotle, he was the first to take a truly scientific swing at it. He used simple morphology, basically dividing plants into trees, shrubs, and herbs, and splitting animals into two very basic camps, those with red blood and those without. Fast forward centuries later, and Linnaeus gave us the famous two-kingdom system, just Plantae and Animalia. Super easy to understand, right? But as you can probably guess, forcing every living thing into just two boxes left a massive number of organisms completely out in the cold. And that brings us to a huge, game-changing pivot in 1969 with R.H. Whitaker's five-kingdom classification.
So why did that old plant and animal system fail? Well, it grouped vastly different things together just because they shared, say, one random trait, like a cell wall. It totally failed to distinguish between a tiny, single-celled prokaryote and a massive, complex eukaryote. But as you'll see here, Whitaker's updated criteria brilliantly fixed that. Notice how we're no longer just looking at what an organism looks like from the outside. We're getting microscopic. We're sorting by cellular structure, the exact chemical makeup of the cell wall, and, absolutely crucial here, the mode of nutrition. Basically, does it make its own food, or does it absorb it? This was a revolutionary upgrade in how we map the biological world.
All right, so here's our roadmap for today's micro safari. We're actually going to skip the familiar plants and animals for now and focus entirely on three fascinating, mostly invisible kingdoms: Monera, the extreme microbes; Protista, the aquatic misfits; and fungi, the unique decomposers.
So, let's head to our very first destination, Kingdom Monera, home of the extreme microbes.
Now, what's really interesting about this slide is how simple, yet beautifully distinct, the shapes of these bacteria are. These guys are the absolute rulers of the microscopic world. I mean, hundreds of them exist in just a single handful of soil. And we generally group them into four main shapes: the spherical cocci, the rod-shaped bacilli, the spiral spirilla, and the little comma-shaped vibrio. They might look simple, sure, but as a group, they possess the most extensive metabolic diversity of anything on the planet.
And within Monera, we find the ultimate survivalists, the archaeobacteria. These bacteria are wild because they thrive in environments that would literally instantly kill almost anything else. Because they have a fundamentally different cell wall structure, they can just shrug off the harshest habitats on Earth. We've got halophiles living in extremely salty areas, thermoacidophiles practically bathing in boiling hot springs, and methogens hanging out in marshy swamps. Those methogens even live inside the guts of ruminants, like cows and buffaloes, where they uniquely produce methane or biogas right from the animal's dung. Crazy, right?
Now, we can contrast those extreme survivalists with the eubacteria, what we call the true bacteria. There are thousands of different types of eubacteria out there, and they're characterized by a rigid cell wall and, if they move around, a little whip-like flagellum. This group actually includes cyanobacteria, those blue-green algae that have chlorophyll and photosynthesize just like plants. Eubacteria are basically nature's ultimate microscopic workforce. They're decomposers, they help make our curd from milk, they produce antibiotics, and the chemosynthetic ones play a massive, absolutely crucial role in recycling essential nutrients like nitrogen, phosphorus, iron, and sulfur.
Okay, before we leave Kingdom Monera, I have to drop a truly surprising fact on you. Meet the mycoplasma. These little guys completely lack a cell wall. Seriously, no wall at all. In fact, they are the smallest living cells currently known to science, and they can survive entirely without oxygen. But, unfortunately for us, a lot of these strange wall-less microbes are highly pathogenic to both animals and plants.
Moving right along to Section 2, Kingdom Protista, otherwise known as the aquatic misfits.
I actually love this quote from the source text. What may be a photosynthetic protist to one biologist may be a plant to another. It just perfectly captures the misfit nature of protists. The boundaries of this kingdom are, well, famously blurry. They share so many features with other kingdoms that classification gets super tricky here. But the one unifying feature, their cell body contains a well-defined nucleus and membrane-bound organelles, which totally sets them apart from the bacteria we just looked at.
And this brilliantly illustrates the sheer physical diversity within Protista. Over on the left, we have the dinoflagellates. These marine microbes have stiff cellulose plates on the outside. It's essentially microscopic armor. And some of them multiply so rapidly that they cause toxic red tides, literally turning the sea red and killing off marine life. But contrast that tough armor with the euglenoids on the right. Instead of a rigid wall, euglenoids have a protein-rich layer called a pellicle that makes their bodies completely flexible. And here's the wild part. When euglenoids are deprived of sunlight, these little photosynthesizers actually turn into active predators, hunting down smaller organisms.
Another massive group of protists are the chrysophytes, which includes the diatoms. These microscopic producers just float passively in ocean currents, but they are uniquely encased in cell walls embedded with silica. They form these two incredibly thin, overlapping shells that fit perfectly together, honestly kind of like a tiny microscopic soapbox. And because silica is virtually indestructible, trillions of these tiny glass-like shells have built up on the ocean floor over billions of years. This creates what we call diatomaceous earth, a gritty soil that we use today for everything from polishing to filtration.
Let's check out two more fascinating protist lifestyles. On the left, you've got slime molds. These are bizarre saprophytic protists that slowly creep along decaying twigs and leaves, just engulfing organic material as they go. If the conditions are right, they can form these massive aggregations called plasmodium that spread over several feet. Now, on the right is the paramecium. Totally different vibe. It's a ciliated protozoan that actively zips through the water using thousands of tiny beating hairs, or cilia. It uses the highly coordinated movement of those cilia to literally steer food-laden water straight into its gullet.
So, that paramecium belongs to the protozoans. They are completely heterotrophic, living entirely as predators or parasites, and biologists actually believe they're the primitive relatives of animals. They basically come in four major flavors. You have the amoeboid protozoans, which hunt by throwing out false feet, pseudopodia, to grab prey. You have the flagellated protozoans, sporting whipping tails, which include parasites that cause awful diseases like sleeping sickness. Then there are the ciliated protozoans, which we just saw. And finally, the sporozoans. These guys have a highly infectious spore-like stage in their life cycle. The most notorious one, Plasmodium, the exact parasite responsible for malaria.
All right, moving into our third and final kingdom for today, Kingdom Fungi, the unique decomposers. This incredibly cosmopolitan group really has it all, containing everything from the savory mushrooms on your pizza to the yeast that makes your bread rise. But, when you look at a mushroom, you're literally only seeing the tip of the iceberg. Except for single-celled yeast, fungi are actually filamentous. Their true bodies consist of these long, slender, thread-like tubes called hyphae. These hyphae weave together under the ground to form a massive, mostly hidden network that we call a mycelium.
Now remember earlier how we talked about the old two-kingdom system failing? Well, for the longest time, fungi were just lumped right in with plants simply because they grew in the dirt and didn't move. So the crucial point is their cellular makeup. Fungi are heterotrophic. They absorb soluble organic matter from dead substrates, meaning no photosynthesis at all. Plus, their cell walls are made of a tough substance called chitin and polysaccharides, while plant cell walls are made of cellulose. The discovery of that unique chitin wall was the exact reason fungi finally got rightfully kicked out of the plant kingdom.
Okay, let's see how all these structures play into their life cycle. When fungi reproduce sexually, it happens in three very specific, distinct steps. Step 1 is plasmogamy. This is where the protoplasms of two motile or non-motile gametes physically fuse together. Next up is karyogamy, which is the actual fusion of their two nuclei. And finally, meiosis kicks in within the zygote, resulting in brand new haploid spores. These spores are then produced in distinct structures called fruiting bodies, which, by the way, is exactly what a mushroom is.
We essentially divide this massively diverse kingdom into four main classes. You have the phycomycetes, like your common bread mold. Then the ascomycetes, or sac fungi, which include valuable stuff like edible truffles and yeast. Next are the basidiomycetes, hosting your classic mushrooms, puffballs, and even parasitic plant rusts. And finally, the deuteromycetes. I love these guys because they are hilariously known in biology as the imperfect fungi. Why? Well, just because science hasn't discovered their sexual stage yet. The minute a perfect sexual stage is discovered for one of them, it immediately gets reclassified and booted into one of the other three classes. Which brings us right back to where we started. You see, classification is never really a finished job. From Aristotle's simple red blood rule all the way to Whitaker's 5 kingdoms sorting by cell walls and nutrition, our map of the biological world has constantly evolved. It really begs the question, as genetic breakthroughs happen and future microscopes let us peer even deeper into the very fabric of life, how are we going to redraw these boundaries next? It's an absolutely fascinating puzzle, and the coolest part is, we're still piecing it together today.